Well, hello, folks. So this is Dr. Ashbaugh. Um, so we've covered the prokaryotes. I've got multiple videos on that, primarily dealing with bacteria. That's kind of the major focus here because those are organisms we can work with in the laboratory. We're outfitted to do that. And from a clinical perspective, they're really important. So it makes sense that we cover those extensively. We'll be dealing with viruses in a later chapter. And while we don't do anything with them in lab, which, I'd like to rectify that and somehow figure out a way to do more with them, but we don't, unfortunately. But there'll be a separate exam dealing with material from the chapter on viruses, which I believe is chapter 6 in Tularo and chess. But sandwiched between between the prokaryotes and the viruses, we've got the eukaryotes. And I've posted one lecture on endosymbiosis theory, this idea that was spearheaded by several folks, primarily Lynn Margulis, back, I guess, in the late 60s, mid to late 60s, and on into the 1970s. They accrued evidence and sort of built that into this framework for the endosymbiosis theory, which is about as good an explanation as we have for the evolution of eukaryotes. Okay, so there's that video that I posted, and what I need now to do is cover the eukaryotes in greater depth. Now, part of this chapter deals with eukaryotic cell structure. And actually, this lecture I'm going to do here in just a little bit relates to that. I'm going to try to keep it brief, though. I'm trying to, it could be hours long if I went off the deep end with it, but I'm going to try to keep it somewhat shorter. And that is because most of you have had some exposure to eukaryotic cell structure, whether you remember that or not. I'll probably jog your memory as I go along and be like, oh yeah, I remember learning that in 7th grade or whatever. But yeah, when you learn about cells in, say, grade school, middle school, high school, or in some of the college classes that you've had, many of you are veteran students and have had A&P 1, for example, maybe had general biology at some point. Those are courses wherein often there's a whole unit on cell structure. And that stuff that you learn there is pretty much the stuff that I'm going to overview here. So let me do that. And then we'll have another lecture following that, which deals with the different groups of eukaryotic organisms. So we'll talk about things relating to those organisms, how they make a living, what their roles in the grand scheme of things are, and so forth. So anyway, let me now go ahead and share my screen. Let me pull up this PowerPoint first. I'm probably not going to use the PowerPoint all that much, but when I pull it up, it lets me duplicate what I have on my computer screen onto this other device that I have, which I can use to draw things and write things. And really, I'm just opening the PowerPoint so I can get access to that. All right, so, this is, if you have Tularo and chess, if you're using that book, which again, you don't have to, all right? You're not obligated to do that, but this is the book from which I'm drawing my information. So this is the heading, I guess, of that chapter. So let me go ahead now and pull up this whiteboard. And I think I'm going to use that more than anything. So first off, I know I'm going to spend more time talking about who the eukaryotes are later, I'm going to go through each of the major kingdoms that contain microbes. But let me kind of introduce what I'm going to talk about today, eukaryotic cell structure, with a little overview of eukaryotic systematics or classification. So we have been talking about, let me increase the size of my lines with my little stylus I have here. We've been talking about members of two domains. These prokaryotic organisms, which were dealt with in the chapter 4, are composed of a couple of different groups that only share some relatively superficial features that aren't shared in common because of genetics. All right. And so there's two groups within the prokaryotes that aren't even that closely related. And so those would be the domain. And let me put the term prokaryotes in quotes because that's not an official taxonomic group. There's no domain or kingdom or class or phylum prokaryotic or whatever. It's just a term of convenience. We lump these groups that share in common the fact that they don't have true nuclei in their cells. And that's basically the only thing really that unites them, other than the fact that they're very small. So one of those groups is called the domain Archaea. And these are the guys that were elevated to the domain level, a new taxonomic category, the domain. They were elevated to that level by Carl Woese, who was a biologist at the University of Illinois. And he used data coming from the study of the ribosomal RNA molecule. Ribosomes from Archaea were extracted. The molecules were sequenced, and those sequences were compared against those from other organisms. And he discovered that, hey, this group is distinct. Well, number one, it's a group unto itself, and it's very different from everybody else. And so despite the fact that we're talking about very small bacteria-like cells that have a prokaryotic sort of structure, there's no true nucleus or true organelles. Despite that, they are a group unto themselves. All right, so these are the Archaea. And then the other group of prokaryotes composes the bacteria. It is the bacteria. That's the domain bacteria. Okay. And that breaks down into bunches and bunches of other subcategories, different phyla and classes and so forth. And then the other major group of organisms on our planet, the one we're going to talk about now, is the eukaryotes. That's a Y. Eukaryotes. And notice I didn't put that term in quotes because the eukaryotes, all of the different members of that group, are united together on the basis of genetics and cell structure. They make up what's called the domain Eukarya. Okay. Domain Eukarya, right? The eukaryotes, the domain Eukarya. And this is a group separate from either of the other two prokaryotic groups. And we consider them special. The term eukaryote means, if we break it down, look at the roots in it, eu means true. And karyo refers to nucleus. So the thing that's special about these members of the domain Eukarya is that they have true nuclei. That is, their genetic material is contained inside of the cell, wrapped with a covering that protects it. And that covering is made out of cell membrane material. It's made out of phospholipid, right? And actually I guess I might have mentioned this when I talked about the evolution of eukaryotes in the little video I did on endosymbiosis. That covering is actually two layered, right? It's not a single membrane, but it's two membranes. Okay, so let's write that down. Nucleus. This is a key word here in our discussion. The nucleus is the genetic material. It's the DNA for the organism, all of its chromosomes. DNA covered by an envelope. We call it a nuclear envelope of two phospholipid bilayers. All right. A phospholipid bilayer is basically just a cell membrane. These phospholipids are molecules that have like a little head made out of phosphate. That's phosphate. And then these little tails are fatty acid tails. They're fatty acids. And so if you have a bunch of these phospholipids, I'll draw them like this, and you put them into, say, an aqueous solution, or just put them in water, plain water, they will arrange themselves so that they form like a skin on the surface of the water. And if we look at that at the microscopic level, it'll have one layer of phospholipids with their heads facing out, and then another layer beneath that with their heads facing in the opposite direction. And that's a membrane, very thin layer, and it's composed of phospholipids. That's a phospholipid bilayer. The covering for a nucleus in a eukaryotic cell is composed of two of those, right? It's a double layered structure. And of course, that's sort of the headquarters of the cell. The DNA, is broken up into segments or genes, each of which codes for enzymes, and those in turn control all of the biochemical metabolic activity in the cell. So all of the members of the domain eukarya have those. In the prokaryotes, the archaea and the bacteria, there's DNA, and it's not covered by anything protective. It's just sort of floating around in there in a little mass called a nucleoid. All right. All right. So there's that, the domain eukarya. And who are the members of that domain? This is important. And I've touched on this stuff in previous lectures, but it's worth going over again. That's why. Let me fix that. Eukarya. So the members of the domain eukarya include the kingdom fungi. And we're going to be talking about those later. By the way, some people say fungi, as I do, and others say fungi. I guess, it makes a whole lot of sense to say fungi. That's, I think, probably the majority of folks say that. I just learned about fungi from people who pronounced it that way. And I know for a fact there are some really outstanding experts on these organisms who say fungi. So either way, however you want to do it, whatever sounds good to you. If one sounds annoying to you and the other one doesn't sound annoying, pick the one that's not annoying to you, but either way, it's fine. But that's a kingdom unto itself, and it contains members that, may not be microbial their entire lives. Organisms sometimes have life cycles where at different stages, they'll have different morphologies and different behaviors and, different sizes. And this is true of the fungi. At some points in the lives of many fungi, they are microbial and they can have important effects on other organisms. At other points in the life cycle of many of these fungi, they're quite large, and so it would be macro, but they are worthy of inclusion in a microbiology course. Things like yeasts and molds and so forth live out much of their life in a way where they're more or less concealed because of their small size. So they're worth keeping. All right. So we're going to study them a little bit later. Another kingdom that includes a large number of microbial organisms is the kingdom Protista. And let me put a little asterisk by this. All right. And the reason that I do that is because this kingdom is very controversial. There are a lot of people who are working hard at categorizing the organisms within this kingdom. People working on what we call systematics, that is the study of relationships among other organisms that ultimately will be used in classifying them. There are quite a few folks who feel the kingdom Protista is really better treated by breaking it up into multiple other kingdoms. So what currently we call one kingdom is really probably more than, say, a dozen kingdoms that are lumped together. But for now, we're going to treat them as just the kingdom Protista. And within it, you have large numbers of smaller taxonomic categories, different phyla. We're not going to get way off into discussing each of those in turn, but we'll just say generally the members of the kingdom Protista break down into those forms that are plant-like, which we call the algae, okay, because they're capable of photosynthesis. They have pigments inside of chloroplasts, which are little organelles that they use for that purpose, and they can gather light, and that energy then can be used to help them make their own organic molecules that they can use for energy. So they more or less, if you want to put it really crudely or informally, they make their own food. So those are algae, and many of those are microbial. Most of them are microbial. Well, not most, but a lot of them. And then we have animal-like forms. Among these protistans, and they are kind of lumped together under this one heading. They are the protozoans. And neither of those names, algae or protozoans, is an official taxonomic category. But any of these members of the kingdom Protista that are incapable of making their own food, which have to go out and ingest other organisms for their nutrition, we would refer to these as protozoans. All right. And there are many of these that are microbial. And so anyway, that's kind of going to be our breakdown of the kingdom Protista. We'll talk about the members of those in greater depth in a subsequent lecture. And then as far as groups that contain microbes go, we have the kingdom Animalia, which in comparison to both the fungi and the Protista contains far fewer microbial forms than those other groups. Okay? Most of the animals are, as you know, macro, very large, relatively speaking. If you can hold it in your hand and, look at it without the aid of a microscope. It's a macro type of organism, and most of the animals are like that. There are some that spend either all or at least part of their lives in a form that's microbial. Okay, so we do want to include them here. So who would be some of the groups of microbial animals? Some arthropods are, like I say, at least for some of their life cycles, going to be small enough to be considered microbial. And they're active and can have an effect on other organisms, either through parasitism or predation or some other way. They can have an effect on other organisms, so they're worthy of inclusion in a course like this. Okay, so some arthropods, those would include things like mites, which are little tiny versions of, say, ticks and spiders. They're in that group. They're arachnids. Mites, maybe some insects could approach a size small enough to be considered microbial. At least the nymphal, the juvenile stages of some insects would be that way. And then among the animals, you also have some worms. The fancy word for worms is the helminths. Okay. Well, some of these, when they're full grown and sexually mature, will be quite large. Again, juvenile stages can be very, very small, and you'll need a microscope to be able to see their features well enough to be able to identify them. And so they're worthy of inclusion here in a course like microbiology. So those are the groups that contain at least some microbes. Okay, those are the groups of eukaryotes that contain at least microbes, some microbes. One kingdom that's not missing would be the kingdom Plantae. So I'll mention them here. Okay. The plants have some stages in their life cycles that are quite small, but generally speaking, the really important stages in the life cycle of these, from the standpoint of importance in ecosystems and so forth, would be the large, sexually mature stages. Plants, trees and so forth. You have a few like mosses and liverworts and hornworts that are kind of reduced in size and smaller, but still mostly they're macro types of organisms. So there are no microbial plants. Okay. So we'll go ahead and kind of just, that's an important group, but from the standpoint of microbiology, there's nobody out there calling themselves a microbiologist who strictly is studying plants. They might study microbes that affect plants, fungal organisms that infect plants, or viral parasites that infect plants, but there are no plants themselves that qualify strictly as microbes. Okay, so those are the things we're talking about. All right, so we'll come back to these kingdoms, fungi, protista, and animalia later. Like I said, I'll do a separate lecture on those. But now let's talk about the general features of eukaryotic cells. If we were to look at all of these eukaryotic organisms, what are some features that really stand out that are hallmark characteristics of their cells? That's what we want to do next. And what we'll do is we'll work our way in from the outside into the interior of the cell, and we'll talk about parts there. We'll start outside and we'll move in. Okay, so the first thing I want to talk about are the appendages that might extend or be attached to the cell that extend out into the surroundings. So eukaryotic cell appendages. Now these, if you recall, when we talked about the prokaryotes, we talked about things like flagella and pili and fimbriae, and within those different categories, there's sometimes variation. So there's quite a lot of different appendages that can be seen in prokaryotic cells. And the eukaryotic cells, it seems that there's fewer kinds of appendages. And those that are there, if they're similar to those of the prokaryotes outwardly, we'll find that inwardly they're very different. All right. So not as great a diversity of different cell appendages in the eukaryotes, but there are some, and we want to talk about those. And the predominant ones that we're interested in, for the most part, are going to be flagella. Okay, eukaryotic cell flagella. As is the case with the prokaryotic flagella, these are longer, sometimes longer than the cell that possesses them. They can exist as just a single structure. A cell might have just one of these, or in some cases there can be a few, and maybe sometimes many. There are some cells in the protista, for example. There's some organisms that are protistans that can have very large numbers of flagella coming off of their structures, off of their bodies. So there's variation in terms of numbers and position and all of that, just as is the case with the prokaryotes. But let's talk about the structure of the eukaryotic flagella. In a eukaryote, the flagellum is composed of primarily a protein called tubulin. Okay. And that's unique to these guys. The prokaryotes have a type of protein known as flagellin that their flagella are built out of. But here it's tubulin. And the tubulin protein, actually, if we were to look at it at the ultra microscopic level, we'll find is composed of these little subunits that are joined together, and they're actually rolled into a tube. So you start off with this chain of subunits of protein that look like little beads, I guess, and that gets rolled into a tube. All right, so it'll look like that, I guess, and it's going to be hollow in the interior, and so that's called a microtubule. And I know my drawing is artistically, it leaves a lot to be desired, but hopefully you get that. You can take this little string of protein, it's like a thread, more or less, made out of these little subunits, and roll that very tightly into this tube. It's like a very small little straw, and we call that a microtubule. And that's what's used to make up the interior of the flagellum. All right. So like, let's say, for example, you've got a eukaryotic cell. Here's a eukaryote, and I'll show its nucleus in there, which is, the standout feature that tells us, hey, this is a eukaryote. It's got its nucleus at one end of that cell. And by the way, the organisms that possess these have to live in some liquid medium, very watery medium. So pond water, Some of them live in the ocean. They can withstand the saltiness of the ocean. Some of them can make their way into the bodily fluids of organisms and be parasites, or, wherever they live, there's got to be abundant water. Otherwise, the flagellum is pretty much useless. The flagellum is used for locomotion, to push themselves through this liquid medium. And so let's say this is one of those organisms, an aquatic organism. And you'll see coming off of it is a flagellum here, all right? And if you were to take and section through that somehow or other and see what that looks like in cross section, what you'll find is that on the outer surface, let me use a different color for this. Let me use red. On the outer surface of the flagellum, where it's cut, all right, there is the cell membrane. All right, so that's a cell membrane. The same membrane that covers the bulk of the cell extends back over the flagellum. All right, so that's a membrane that covers it. And then in the interior, we have microtubules, all right? And when you cut through a microtubule, you cut through this like that, you'll see that it's hollow in the center. It's going to look like a little circle. And so what we'll see inside of here are these circles, right? Each of which is a microtubule. Notice there's a pair of them side by side here. Those are two microtubules, all right? And then we go a little further and there's going to be a couple more. And then, so they're arranged in pairs here. And notice they're going to be right underneath this membrane. And if we look at all of them and count them, we'll find that there's actually 9 pairs of microtubules surrounding this thing, all right? So I won't draw all the pairs in, but there's 9 pairs on the outside. Nine pairs outside. That is, they're right underneath the membrane covering, but they're outside of this very center of the cell. In this very center are a couple of microtubules that they look like they're paired up, but they're really not. They're separate. They're just stuck side by side there. Notice I drew them with a little bit of space between them, right? So you got 9 pairs outside, and you've got these 2 in the center. 2 in the center of the flagellum. That's a 9, right? 9 pairs outside and 2 in the center. So we refer to this arrangement of microtubules of tubulin as a 9 plus 2 arrangement of microtubules. So you can see they make a nice little sort of skeleton for the flagellum. You know, they're going to be long, and they're going to provide the framework that's going to form that flagellum, giving it its, thin shape, its elongated shape. Now, in addition to the tubulin, we have, coming off of these pairs on either side, another protein. So let me advance this, and I'll show this here. Here's our cross section of the flagellum again. Let me see if I can put 9 pairs in here this time. There's 3. Hey, there we go. So there's the 9 pairs for the outside. And then here's the 2 in the center. All right. So this is the cross section through the flagellum. And coming off of the sides of these is another protein. Let me use a different color. I've got these cool little, got a little palette here of different colors I can use. So coming off the sides of these proteins is another protein. Okay. It looks like a little hook. And these are going to be arranged all down the length of the microtubule. You're going to have these little arms coming off, in tremendous numbers all along the lengths of these microtubules. So this is just that one little position there. There's a little arm that comes off. And then here's another arm here coming off of that neighboring microtubule. And that's just going to continue on around here. I know my drawing's not the best. Notice how those little arms are kind of clasping together, like two hands hooking together, right? They sort of overlap. These proteins are called dynein. So this is another protein. These are called dynein arms. Okay. And all of them are going to have those. And these are not temporary structures. These are actually, permanent fixtures of each of these microtubules. And I know my drawing is, again, not the best, but I'm in a hurry. And so there are those dynein arms. And what's more, there are dynein arms that reach out and connect to these guys in the center as well. So I won't draw them all in, but you get the idea. There's interaction between the microtubules in the surrounding portion of the flagellum structure. And then there are dynein arms coming off of these tubulin microtubules in the center that overlap with them as well, dynein arms. And what they can do with the help of ATP that provides energy to make them move and work, they can clasp together and pull on each other and then release. And by doing that alternately, very rapidly, it imparts movement to the flagellum. The flagellum can kind of undulate. And that's how then they're able to move. They can use that flagellum in a variety of ways. They can swish it back and forth like the tail of a fish, and that can push against the surrounding water, which is dense enough that it's going to help to propel the cell forward. Some of them can take the flagellum. Some, say protistans, for example, can extend the flagellum frontwards and twirl it like a propeller and not push themselves, but pull themselves, right? Some of them can extend the flagellum if, say, they're down on the bottom somewhere of the body of water that they're in. They can extend the flagellum and kind of use it as a pusher to kind of drag themselves along. They can use it that way. So there's a variety of ways of using the flagellum. One thing for sure is that where the flagellum anchors to the cell, it is fixed in that position. So unlike what we see in bacteria, where the flagellum can rotate 360 degrees, because it's not, got a fixed attachment to the cell, these guys can't move that portion. They can't twirl this thing. And so the idea of runs and tumbles and so forth, like we see in the prokaryotes, in the bacteria, that concept does not apply here with the eukaryotes. They don't exhibit that difference in forward movement depending on whether the flagellum is rotating clockwise or counterclockwise. That's not something that applies with the eukaryotes. But anyway, that's eukaryotic flagellum structure. And so that's basically an organelle. It's an appendage that is composed of these tubulin microtubules arranged in a 9+2 fashion. And these are moved relative to one another with the help of these dynean arms. Okay, so those are flagella. And like I said, generally speaking, they're relatively few in number, although there are some exceptions to that. And they're quite long in most cases. If you laid them down next to the cell that they're attached to, they may be as long as or longer than that cell. Now, there's another type of appendage that is much smaller that, in fact, retains this 9+2 arrangement. And so the second type of appendage would be a cilium. That's the singular. The plural is cilia, right? And these are nowhere near as common as flagella, but there are some aquatic members of the kingdom Protista that are basically identified on the basis of the fact that they have lots of cilia. All right, there's one group in particular that possesses quite a large number of cilia. It's not the only group that has them. There are other kinds of eukaryotes, including humans, that have ciliated cell surfaces. But in terms of different groups of organisms, there's this one group of protistans, and they are called the ciliates. These are protistan, actually they're protozoan, all right? Remember, the protistans are composed of plant-like and animal-like forms. So these are protozoans, because they're animal-like, that are typified, which means, this is a common feature that they have. They're typified by possessing cilia. That's typical for them. Okay, they're typified by the possession of cilia. And cilia are never usually found as singles. They're not found in small numbers. In flagella, that's pretty common. In the case of cilia, if you're going to have them, typically you're going to need a lot of them. And so, this is a really interesting group of protozoans. As is the case with most protozoans, they are single-celled, right? And a very common one, actually a genus, which includes several species, a very common collection of these are members of the genus Paramecium species, let's say. There's multiple ones of them. There's one called Paramecium caudatum, another one called Paramecium micronucleatum, and so on. These are very commonly studied, mostly freshwater ciliates that live in pond water and things like that. Some of them can actually be clinically significant, especially as infectious agents in pigs and horses and some other animals. And humans are usually not pathogenic, but paramecia are typically sort of slipper shaped. All right, so they're elongated, sort of oval. They'll have like an indention on one surface that leads into, you can see this inside of them. They're small enough that you can actually see inside of them. There's this kind of funnel shaped area, and they'll have some little protein fibers that kind of form a little framework around it. And at the end of this, you'll see little bubbles of membrane being produced as food items. So these are food items here. As food items are taken in, they enter this structure called the oral groove and then pass down into this basically indention, this tube that runs in. And then these are little food vacuoles here that are forming by endocytosis. Almost said exocytosis. So food vacuoles, and then those go into the cytoplasm of the organism, and enzymes then act on the food within those vacuoles and break them down. But so that's kind of how they make a living. Now they move around in the environment very rapidly with great agility because they have over their surfaces very large numbers of cilia. And they even have cilia around this oral opening here. All around it, they've got cilia that they can use to help draw food in. And then they've got the cilia here. In fact, over the whole body, you're going to have rows of these cilia, these little hairs. Now I'm cluttering my diagram up a lot. I probably should use different colors, but they're entirely covered by cilia. And the cilia can beat rapidly. And when they do, they usually beat in a coordinated way. It's not like they move one cilium one way and then its neighbor in a different direction. They usually move in the same direction. And if you can actually see this happening, and there are microscopes powerful enough to afford this ability, as you watch them move, when they beat their cilia, they move in waves. So the cilia that are, say, beating at this point will beat first, and then that basically progresses back from there all the way to the other end, and then it'll start over again. So it's like watching the wave at a football stadium. There are waves of ciliary movement, and they can, if necessary, switch the direction of the wave. So if at one moment in time the wave is beating, in one direction, they can reverse that and go in the opposite direction, right? They can turn very rapidly. So they can spiral through the water column. They can do a variety of different movements and change between those movements basically instantaneously. There actually is a framework of proteins underneath the covering of the paramecium, underneath its membrane. It's called an infraciliature. You really don't need to know that, but what it does is it connects them internally. It's like a mesh or a grid system of little proteins that unite the bases of each of these cilia. And that's how it is that you can coordinate them. When when one cilia moves, it kind of moves that infraciliature so that the next row of cilia are going to be affected and they'll move. And it just, proceeds from there. So these are among the more agile, more rapidly mobile small organisms that have yet been discovered. Oh, and they do have a nucleus. So let me draw its nucleus in here and just mention really quickly something interesting about the ciliates. This is kind of just additional information. They actually have two nuclei. They have what's called a macronucleus. And we'll talk about these in depth, not really gory depth, but we'll talk about them in some depth later. But for now, just know that there's actually two nuclei in most of these species of paramecium and other ciliates. There's a macronucleus, which obviously the big one, and then a smaller nearby micronucleus. And they'll use the micronucleus when it comes time to undergo pairing with other individuals for the purpose of reproducing eventually. There's a phenomenon known as conjugation that we'll talk about that involves use of that micronucleus. Anyway, that's a feature that tells us that these are good solid eukaryotes, right? And they have cilia, so they are ciliated. And there's many different genera of these. There's one called Vorticella and one called Didinium and one called Stentor and so on and so forth. Things that if we were to go down to the Jackson State Pond and draw out some water, we could put that under the microscope and see a lot of those. And, maybe if we have some time this term, we'll do some of that, but we're usually burdened with so many other things we don't really get to it, but they're very common. All right, uh, so we have flagella and we have cilia. Those are two of the kinds of appendages that you'll see in some of the eukaryotic cells. All right. So moving on from there, let's move on to the next thing. So now we want to talk about the actual covering of the cell, the cell envelope. And the cell envelope is the covering of the cell. And in eukaryotes, there's some special things about the cell envelope. Recall that in bacteria, there is a definite, well, there's a glycocalyx. So let's make mention of that here. Eukaryotes have that too. There is a glycocalyx. And let me number these because there's a few things anyway, and keeping them straight is easier if I number them. So the glycocalyx, this is the outermost covering. All right. And it's composed mostly of, again, as was the case with the prokaryotes, carbohydrates. Composed of carbohydrates, sugars, and so forth. And also there'll be some protein in there too. Okay. So things like glycoproteins. That's a combination of some small, say, chain of sugar and some protein embedded in the outer surface of the cell. So some glycoprotein may be there. Glycolipid, which is sugar and lipid together. All right, et cetera. Those are some of the things we might expect to see in a glycocalyx. And this is the outermost covering. This is where you're going to have your cell surface antigens. Basically markers. That can be used to receive substances. If there are chemicals produced by one cell, they can be released into the surroundings, and these antigens can bind those. They can be used for cell-to-cell recognition. And so in there are receptors. And the glycocalyx can be protective, can have a protective function, like serving sort of as a capsule. Okay, so there's a variety of things that glycocalyx can do, all right? And so this is not considered an organelle, but it is an outer covering that can be valuable in a variety of ways, functional in a variety of ways. So there's going to be a glycocalyx. On your cells, for example, you've got a glycocalyx, and it is composed of these same things. So the markers on your cells that identifying your cells as belonging to you, are the same markers that exist on other cells. Say, for example, you got a transplant from some other individual, you received that transplanted organ or tissue. If the glycocalyx markers are different enough on that tissue that you've received, your immune system will respond to it as being foreign. All right. So cell-to-cell recognition and tissue compatibility, histocompatibility, is determined in humans by the glycocalyces. So that's a layer that's always there. Okay. And then the next layer below that varies depending on the organism that we're talking about. If we're talking about a fungus, if we're talking about an algal organism, in those two groups primarily, maybe a few protozoans, there's what we call a cell wall, okay? So cell wall is the next layer in, and it may be present, okay? Some organisms don't have them. These are found in algae. They're found for sure in fungi, and maybe a few protozoa, although protozoans being more animal-like, they tend not to have them. All right. The animals never have cell walls. Okay. The whole organism, animals are multicellular. The whole organism may have a covering that it secretes that's protective. But if we look at the cells that are making up, say, the epidermis of the organism, they're never going to be like those of these organisms. They don't have cell walls, but these organisms have them. All right. And the composition is variable. I didn't really mention function yet. And let's just say, rather than belabor the point, let's just say the cell wall, if it's present, is protective. I mean, it's sort of self-evident that that's the case, right? These are really protective. They help the cell from, swelling and bursting under certain conditions, or maybe help the cell resist mechanical tension that it might be placed under, mechanical stress. But yeah, they're protective. Cell walls are protective. All right. We don't have to go any further with that. But in terms of composition, there's some interesting stuff there. So the composition varies. Okay. In fungi, fungal cell walls are made of chitin, this substance called chitin. Let me underline that. It's a key word. So chitin, not pronounced chitin. I've had students who have gotten study guides or something and have seen that word and maybe weren't in class the day I talked about it, and they would pronounce it chitin. It's actually chitin, all right? And chitin, rather than just really going into a overly extensive coverage of it, you could think of it as sort of a mixture of some carbohydrate along with some components that would be expected to be in protein. So there's some amino acid in there, there's some amino acid subunits, and then there are some carbohydrate subunits together that make up the chitin. So it's a very interesting substance. It's found in other groups of organisms. We have chitin in the cell walls of the arthropods. So insects and crustaceans also commonly have lots of chitin in their bodies. So fungal cell walls are composed of chitin, and or there's some oddball ones that contain things like sterols. Cholesterol-like molecules would be present there. They could be present in cell walls. Also, many with some cellulose, in addition to the chitin. Cellulose may be present too. And so cellulose is a very common component of plant cells. It's what makes up the cell walls of plants, and is the reason why plant material, when we ingest it, is not easily digested. we can ingest it, but digesting it chemically is very challenging because of cellulose. Cellulose is composed of glucose molecules that are polymerized. That is, they're joined together in big, long chains. And the way that those chains are built and the way they branch makes it very difficult to digest that material. So that can be protective as well. So, but chitin is the premier cell wall component of fungal cell walls. And then some species have, some cellulose, I guess, helping to reinforce that. So those are fungal cell walls. Let me put a letter by that. And then we'll go over here to B, which would be algal cell walls. That's the other group of microbial eukaryotes that have cell walls. And so these can be a variety of things. They can be composed of cellulose. They can be composed of what's called pectin. Okay. They can be composed of, pectin, the same stuff that we get from certain plants that's used as a thickener, say in making jelly and that sort of thing. Pectin is an actual cell wall component in plants and algae. So pectin can be there. See what are called glycans, which are more complex carbohydrates in addition to cellulose. And then weirdly, there's some of these algae, in particular the diatoms, they have what's called silicon dioxide. Okay, this is silicon dioxide, which is the principal material of glass. And it's the principal component of the substance we know as glass. And this is in the diatoms. These are single-celled algae that live in both fresh and salt water. And they're commonly found, some of them can suspend in the water column, but others will settle out on rock surfaces and so forth. And they have in their cell walls, they've got these little glass shells, essentially, that are finely sculpted and very beautiful, very geometrically complex. And actually the sculpturing of the cell wall can be helpful in identifying the organism. So some of them are shaped like little boomerangs, sort of like this. And then you'll have others that are shaped like this. There's a whole slew of different shapes. Some of them are just really long, slender cells like this. They're microscopic. You'd have to have a microscope to see them, but typically what they have are two halves to this covering, and there'll be a split between them. So they fit together like clamshells. And you'll see, let me use a different color. Let me use black. You'll see these little striations, these little sculpturings, little grooves that seem to be cut into the surfaces of these cell walls. So that's a common shape and distribution of these. All right, so they've got these little slits that are cut in them, or sometimes there'll be little pores in there. It just varies. And they're able actually to extend some cytoplasm out through those slits, and they'll have sort of like a covering outside of that cell wall of this gelatinous material that's from the interior. But when they die, all of that perishable, decomposable stuff within them basically degrades, and what's left behind are these little cell walls, which look like little shells, and they can be fossilized. We can find them fossilized. They can accumulate in very, very large amounts that can actually be mined. And so there are places where, ocean water used to be that's receded and it's left behind layer upon layer upon layer of just untold trillions of diatoms that have died. And you can mine that, then go in with bulldozers and scoop shovels and stuff and get it, and they sell it as diatomaceous earth, which some of you may have purchased. It's sort of powdery stuff that you don't want to breathe it in because it's made out of this glass, and so it can get in your lungs and things, but they use that for different purposes. In fact, they used to put it in toothpaste and use it as a tooth polish. But anyway, so there's silicon dioxide in cell walls of some organisms. So there's a variety of different cell wall components that we'll find in the algae. Like I said, some protozoa probably have cell walls. My guess is that they're probably going to be composed mainly of carbohydrate, the few that have cell wall or cell wall-like forms. But those are the two major groups that have cell walls. The algae and the fungi are cell wall possessing eukaryotic organisms. So now the next layer in, and we'll talk about briefly. Here's this little pop-up thing that came up on my screen. Will be the cell membrane. Okay, so we've gone from the appendages on the outside, the flagella and cilia, the cell wall if it's present, and then the next layer of the cell envelope, number 3. And I apologize for using different colors. I'll just keep this color for now. The third layer would be the cell membrane. And this is a fixture. So this is something that you'll find in any cell. We could say that this is an obligatory, uniformly possessed structure of all cellular beings, whether they're prokaryotic or eukaryotic. They've got to have a cell membrane. So there's no variation here in the possession of the cell membrane. There might be some variation in terms of how it's put together exactly. Archaea probably has some different features in the cell membrane than all other organisms, but it's true that they all have this. And so this is a semi-permeable boundary. Okay. And it's composed mainly of phospholipids. In a bilayer. Okay, so the membrane will have phospholipids in its outer surface, which, remember, have these little bead-like heads composed of phosphate. So this is a phosphate head, has negative charge. And then fatty acid tails, which are basically just long tails of carbon after carbon after carbon atom joined together. And then hydrogens are going to be bound to those carbons. And so the phosphate containing part is hydrophilic because it's got a charge, got a negative charge. So it can mix with water well, it can interact with water well. The fatty acid tails are hydrophobic. So that part resists water. And so if you throw these large numbers of them into water, they'll arrange themselves with the heads facing out and the tails facing in. So here's one surface of the phospholipid bilayer, the tails go this way. All right. And then on the other side is a layer that's built the same exact way, but it's just oriented opposite that of the one above it. And so the tails are like this.
There it is. There it is.
All right. So the tails are sort of sandwiched in the center, and that's how these cell membranes are put together. By the way, they're also sometimes referred to as plasma membranes. So if you ever see that, you know that that's what they're talking about. So that's the bilayer. Now, for quite a while after it was discovered that membranes had phospholipids arranged in a bilayer, went back into the 50s and 60s, it was considered to be sort of settled that that's how all membranes are. But what we now know is that they actually contain large numbers of other substances. And instead of just being just a plain pure phospholipid bilayer, really what describes them best is the fluid mosaic model. That's currently the favored model or structure of these things. The fluid mosaic model or theory about how these membranes are put together. I guess they're able to actually literally freeze a cell membrane and then with a micro chisel fracture it and look and see what's in it under electron microscopy. They can see things at that level. It's fascinating that they're able to do that. So instead of just this phospholipid bilayer, you have arranged in it a variety of other substances. So let me just take a little chunk out of my picture here, and I'll draw in something else. Let me use a different color, a little color happy here. So you'll often find scattered throughout the membrane big bulky molecules sort of stuck in there that are embedded in one surface or the other of the membrane. Could be sometimes that they're actually found all the way through. Some of these are solid. Some of them actually have a hole down through the center. So, have a solid surrounding area, but there can be a hole through them. So those are embedded proteins, right? Those are proteins. So that's another thing you can expect to find in the cell membrane. You got your phospholipids, and then you got proteins like this. And those proteins can do a whole lot of different things. They can serve as, let's just list some of these possible things out. They can serve as channels to allow substances to come and go, right? Remember I said this, the cell membrane is semi-permeable. What that means is it'll allow some things to cross it, but others are going to be impeded and they're not going to get through. So a channel like this could let something through, let's say water. Water is not something that readily can cross cell membranes all that easily. But if you build channels like this, water can cross. Or ions, things like sodium and chloride and potassium, those kinds of ions, because of their charges, have a hard time getting through membranes, but channels can be set up to allow them to pass through. So that's one possibility as a function for proteins. Others can serve as receptors. So receptors for things like neurotransmitters, receptors for hormones. In the case of microbes, they can produce chemicals that a cell can release, and it can be used to communicate with other organisms. There's this phenomenon known as quorum sensing, where microbes can all sort of, through communication at a chemical level, can come together to form things like films, biofilms, the plaque on your teeth. a layer of a biofilm, a sort of slimy layer of, say, bacteria growing along the walls of a pipe or something like that. Those form by communication between cells, and receptors are used for that. So you can have those, you can use them as markers, so cells can recognize each other for cell-to-cell recognition. You know, there's a variety of things that these proteins can be implemented for. Some of them are used as enzymes. Let me write that down. Let me make sure that that's in there. Some of them are used as enzymes. So chemical reactions can take place right there in the membrane, you know, and I'm sure if I brainstormed, I can come up with some other things, but you get the proteins in there. And cells can make more of these if necessary. If there's one that's especially valuable for a particular environment that they happen to be living in, they can make more proteins and embed them in there. And so they come and go. They can move around. The phospholipid bilayer is not static. And so these things sort of drift around in that membrane from place to place. That's why we call it fluid, right? And so now you can see more why we're calling it mosaic as well, because a mosaic is something that's composed of multiple parts, which vary in terms of size and shape and how they look. If all the parts were exactly the same, it wouldn't be a mosaic. This is a mosaic. Let's see, what else might you have in there? Besides proteins, you can have sugars. Okay, so let me use a different color for sugars. Carbohydrates. I'll use blue. So here's a chain of sugars. They're attached to the phospholipids here. Here's another sort of chain of sugars that can be formed. I'm showing them looking like little Y's. It's not necessarily how they always are. But this is a chain of carbohydrate. So those that are attached to the proteins form what are called glycolipids. Glycoprotein, rather. I'm sorry. That's a glycoprotein. And then this right here, because we've got a phospholipid bound to a chain of carbohydrate, that's a glycolipid. And so these are very often used as markers that identify cells as being one type and different from others. All right. So maybe for organisms to recognize one another, they'll have these markers to be able to signify that this is who I am, little adornments that they wear that help them to do that. So there can be some carbohydrate in there. So we got phospholipid, we got protein, we got some carbohydrate. And then a final thing that you might see, and I know my diagram's getting really cluttered, but a final thing you might see are what we call sterols. And cholesterol is the chief one of those. And I'll draw these little sort of S-shaped structures in here, little zigzaggy little Z's in here to show those. They may not be scaled properly in terms of size, but what each of those are, these are sterols, a sterol molecule, the chief one of which is cholesterol. But those may be in there. You can have sterols. And their function primarily is to add some strength and rigidity to the cell membrane. So if the membrane's too delicate and it can tear easily, there can be some genes expressed that lead to more sterol formation. Those can be incorporated into membranes to make them a little more rigid, a little tougher. And so that's another thing that you can expect to find in membranes. So let me advance this. So membrane components. Mostly phospholipid. Protein. Okay. The very wide range of functions for those. You've got carbohydrate. Either in the form of glycolipids. I don't know why I wrote it out like that. Glycolipids. Yeah, the glyco root in here, that refers to something that's a carbohydrate composed of sugar, basically. And then we have glycoproteins. And then the final thing that we would expect to see is sterols of different kinds. And the chief one of those is cholesterol. All right. I'll put an exclamation point there because that's the main one. And these are lipids as well, that are used for reinforcement. Those are your main membrane components. Those are the big four, basically. And like I say, these can be positioned and moved around in relation to one another. The composition of the membrane doesn't have to stay fixed. It can vary somewhat, depending on the needs of the cell. All right. So when we call them now a phospholipid bilayer, we need to remember that that's not all that's in there. They're actually a composite of several things. So the fluid mosaic model is the current theory as to how these membranes are structured. And there's liable to be some more things added to that as we go along, but for now, that's what we know about how these membranes are built. And so when we look at cell membranes in bacteria or plants or protista or animals, there's a lot of uniformity there. And it kind of signifies just how important the membrane is to all living things. Okay, so there's that. So that's the envelope. Now, which is composed of the glycocalyx, maybe a cell wall. There's some groups that have those. And then for sure, a cell membrane is going to be there. And those all help to form a boundary around the cell and to maintain its size and its shape. And, to help regulate what comes and goes, to make sure that the interior of that cell is composed in such a way that it'll support the cell's activities. All right, so moving on from there, now we're going to talk about the internal components. We're going to talk about what's in the cytoplasm. Okay. And let me go back. I'll use a different color here. So now we're going to talk about the cytoplasm. This is the interior of the cell. So the outer surface is the envelope, the cytoplasm is everything within, and it's composed of generally two things. It's going to have, number one, cytosol. Okay. And it's going to have organelles. Since we're talking about eukaryotes, we can talk about organelles. And other suspended particles, let's call them, that aren't strictly speaking organelles. For something to be considered an organelle, it's got to have membrane of its own. And there are some things that are important that are in that cytoplasm that are not membrane bound. So organelles, these are all membrane bound. You need to remember that. The same kind of membrane that's in the cell membrane that surrounds a cell is used to make the outer coverings of these organelles. These particles that I'm talking about here are not membrane bound. So you have both kinds of things in there. What is the cytosol? Well, the cytosol, the first thing is really basically it's just water with dissolved molecules in it. And so what are those molecules? The dissolved molecules are proteins, soluble proteins. Okay. These are mostly enzymes. Some can serve as pH buffers that help to regulate pH. Some of them are osmotic buffers that help to regulate water balance in the cell. The more proteins you have in there, the better the cell is able to hold on to water. So those are some of the reasons that the soluble proteins are there. The chief ones, though, are these enzymes, and they control all of the biochemical reactions that are going on in the cell. So you'll have large numbers of those suspended there. You'll have, ions. You'll have starches in some cases, lipids, etc. Molecules that are being used as food, for example, sugars. I didn't mention those, but those are going to be present there in the cytosol. All right, so that's principally what that is. When you remove all of the larger particles from this cytoplasm, the liquid fraction that you have left over is the cytosol, all right? And it includes these things. So now then we can talk about the other fraction, the things that are suspended in there, the organelles. Okay, and I'm not going to do a comprehensive list of these, but I'll talk about the biggies and mention a few highlights of each and then move on. All right, so the chief one of the organelles is the nucleus, obviously. All right, so this is where it contains the DNA, covered by envelope, nuclear envelope. And that DNA, of course, is where the genes are that code for all of the different major enzymes that the cell is going to utilize. And those in turn carry out the metabolic reactions of the cell. So, and that's what determines how the cell is going to function, how it's going to look, all of those different features that it possesses. So the headquarters basically of operations is that nucleus. All right. And I'll put a big star over this one because remember, that's the feature that tells us whether it's a eukaryote. This is the hallmark characteristic of all eukaryotes. All right, that's it. All right, the nucleus. So they got to have that. All right, so that's the first one. Now that envelope, that double membrane that surrounds it makes up what's called an endomembrane system, which is composed of several key parts. Oh, I added another little loop on my M there. Okay, so an endomembrane system. And so let me list the parts of this. So there's what we call a rough ER. There's a smooth ER. And then the other main component of the endomembrane system would be the Golgi apparatus. So these are very extensive structures that are going to take up quite a lot of the volume of the cytoplasm, and they're composed primarily of membranes, right? And really, I should probably list the nuclear envelope as, you know, an honorary member of the endomembrane system, because it is the source of, well, at least the ER, but the rough and the smooth ER basically are extensions of the nuclear envelope, right? So that's why I'm going to include it here in the list of things. So let's look at these things in turn. So let's start off with the rough ER. The ER here stands for endoplasmic reticulum, which is a fancy word that when students sometimes encounter words like this, they immediately withdraw and they think, you know, I'm never going to understand that because it's too technically difficult, but it's not. Endo means inside. Plasmic refers to cytoplasm, right? So that tells us where this is. It's in the cytoplasm. And then reticulum is like a network. So this is a network of membranous sort of sacs that balloon off of the nuclear envelope, right? And it's rough, well, for reasons that I'll disclose here in a second. So let me just draw a little kind of, you know, here's a cell, right? Just some generic cell, and here's our nuclear envelope. Here's the material inside of the nucleus. I'll just kind of shade that in to let us know that there's stuff in there. We call this, by the way, the stuff in the interior, we call it chromatin. That's DNA, but it's more than that. It's protein that helps to package it. That's what we call the DNA when you know we see it during the height of the cell's activity. While it's not dividing, we can see that there's this darkly staining material in there, and that's the chromatin. That's DNA plus protein. That's what it looks like when the cell is active. And then ballooning off of this envelope, and we have phospholipid bilayer material that balloons out and forms these big sort of sacs, right? Well, this is a series of little flat sort of sacs, all sort of connected together, that are produced as an extension of that nuclear envelope. Let me just draw some more up here, a little bit more. And because I'm drawing this in 2 dimensions, it makes it really hard to really show it accurately. But that's how it looks. It's a bunch of flattened sort of sacs of membrane. And, you know, they've been sliced through here, so we're looking into them, but that's how they'll be. And the rough ER will have, studying its surface here, Let me go ahead and put different color. Let me use green. These little green dots all over the surface of the membranes here. Okay. Notice the whole ER is not going to have those, but large amounts of the ER will have these little dots that stud the surface of those membranes. And when you see ER that looks like this, you know that that's rough ER. That's the rough ER. Okay, and then the membrane that doesn't have that, the stuff out further out here, is the smooth ER, right? So let me go back to my other color here. So this area here is rough ER. Down here in this picture here, that's rough ER. And so, and then the area that doesn't have those, all of this, is smooth ER. Okay. My diagram got cluttered real fast. So that's ER. And now you can see why I included the nuclear envelope in the endomembrane system. And so the rough ER in there is rough because it contains ribosomes. Right? So rough ER contains ribosomes. Okay. Whereas the smooth ER doesn't. It's pretty much the same stuff as rough ER, but it doesn't have ribosomes, right? And by the way, there are other ribosomes in this cell. Remember I said the cytoplasm consists of some things that are suspended in that cytosol, you know, that aren't, strictly speaking, membrane-bound organelles. Those are free ribosomes that are just sort of floating around. So you got some of those, but you also have ribosomes that are attached to the ER itself, the rough ER. So that begs the question, what the heck are these ribosomes about? So let's deal with that now, and then we'll come back and we'll talk about the smooth ER. So ribosomes most in a eukaryotic cell are going to be attached to rough ER. Some are suspended, some are free, and thus are suspended. They're kind of floating around in the cytoplasm. So what is a ribosome? Actually, it's a type of organelle without membrane. Some people would actually not classify them as organelles, but this is a non-membrane type of organelle, and it's composed of two parts. It's got a small subunit, so like a little oblong kind of mass of material there. That's the small subunit, and then a larger subunit. Okay. So a small and a large subunit. And these are both composed of the same stuff, composed of primarily RNA. That little R stands for ribosomal, and then RNA means ribonucleic acid. You know, the stuff that's made out of nucleotides, just as DNA is, except the the sugars are ribose sugars, not deoxyribose sugars. So yeah, rRNA, and this is coded by genes. Genes actually are expressed to make this, and that's then formed into these subunits. And these two subunits, when they're joined together, they're not always joined together, but when they join together, they're active and they serve as the sites for protein production. Okay. And so let me write that down here in caps: used for protein synthesis. Exclamation points. That's super important, because if you don't make proteins, you're not going to have enzymes, you're not going to have metabolism, you're going to die. All right. So, and the other thing, of course, proteins are used as structural building materials, and those are important, pretty important too. I think that's an understatement. But anyway, that's what ribosomes do. The messenger RNA that's produced in the cell during translation of the genes travels out to these ribosomes, attaches right in this little groove here between the large and the small subunit. And as that messenger RNA gets pulled through, the information is more or less transduced, and a protein then is formed as a result of that. All right. So like here's a small subunit, here's a big subunit. Now they're joined together. Okay. And a piece of messenger RNA, which is produced by transcription of genetic information. The messenger RNA, this is mRNA, can attach and now then can be passed through this ribosome. And as it's pulled through, a protein is produced using that information. So I'll show a little squiggly thing here. As this messenger RNA goes further and further through that ribosome, this protein here is going to be produced, and eventually, as the messenger RNA reaches the end of the message, the protein is finished, and now it can be used for whatever purpose it's needed for inside of the cell. So that process right there is called translation. Of messenger RNA. And the ribosome is central to that. It's one of the main things that makes that happen. No ribosomes, no translation, no protein production, right? And so that's incredibly important. So if we go back to my drawing of the cell here, the rough ER where those ribosomes are located is a very important center of protein synthesis. So lots of proteins are produced here. And then what they'll do is they'll be passed on up through the ER and packaged. And what you'll see budding off of this thing out here, let me show them here, are little bubbles of membrane. That ER literally kind of balloons out and forms these little bubbles, and inside of each bubble is protein. So I showed proteins earlier looking blue. So there's proteins that were produced back in the rough ER that are now packaged in these little bubbles of membrane. And the proteins can be delivered to other places if necessary, or released into the cytoplasm. There's a variety of things that can happen with them. By the way, these are called vesicles. Okay, so the ER is budding those off more or less constantly. Okay, so rough ERs for protein synthesis. What about the smooth ER? Smooth ER, lacks ribosomes, obviously, so it's not going to be helpful in producing proteins. But instead, the smooth ER is the site of much lipid synthesis. So that would be phospholipids for membranes. That would be cholesterol. Well, that's also used for membranes and other purposes, used for making hormones. So hormones, etc. Just fat in general can be produced there. So when you see a lot of that smooth ER in the cell, you know that that cell is probably adapted more toward production of lipid. Most cells are going to have some ratio of both of the rough and the smooth types. Yeah, so that's important. The enzymes for making those things are contained in the smooth ER. So we got a lot of lipid synthesis going on. And then the last thing that I mentioned was the Golgi apparatus. So if we have synthesis of substances in the smooth ER, the Golgi apparatus can serve as kind of a processing center that can modify those substances. For modification of materials from the ER. And so let me draw another cell here. So here's draw a great big one. So here's our nucleus. Here's some ER. I won't make it super extensive because I need room to draw my Golgi apparatus. So there's our ER. Here's rough. And then there's smooth out there. And then we're going to have the Golgi apparatus. And so what it is composed of are, again, sacs of membrane. They're like pancakes of membrane stacked on top of each other. And notice it's not actually attached to the ER. Okay, so that's the Golgi apparatus. And I guess you should capitalize the G because this is a person's name. There was some person named Golgi that identified this. So these vacuoles, or vesicles rather, bearing substances that were produced in the ER, these vesicles will sort of move toward the base of the Golgi apparatus where they'll fuse and they'll release their materials in to the Golgi apparatus, which then they'll just pass on through there and they'll come out this other side here, modified. So you can think of it as like a factory where you get some raw material that comes into the factory, say, pieces of copper coming into Delta faucet and, big chunks of copper. And by the time that metal goes through all of the processing that goes on at the factory, at the end of that, you get a shower head or, some other metal containing substance or structure. So that's what happens here. Raw materials from the ER are passed through the Golgi and they're modified. All right. And so from there, those vesicles, they can travel to other parts of the cell and have their function there, or they can travel to the cell membrane and fuse with it and release their substances to the outside. So there can be secretion of materials that way. This is called exocytosis. That's where there's this release of substances into the surroundings from the vacuole that's fused with the cell membrane. Okay, so those are the components of the endomembrane system, and incredibly important features of eukaryotic cells. Of course, prokaryotes are going to have nothing like that. A prokaryotic cell is going to have ribosomes basically just free floating as they are here. Another thing is the ribosomes of the prokaryotes are a little different in terms of the size. Prokaryotes have what are called 70S ribosomes. The ribosomes that are found here in the eukaryote are larger. They weigh more. And so they are 80S ribosomes. So there's a little difference there in size. But in terms of their composition, they're the same. They have rRNA in. So that's the endomembrane system. All right, so moving on, I'm just going to go ahead and knock this out. I know it's a long video. What are some other things you would expect to find in the cell? Another thing that you would definitely expect to find in all eukaryotic cells is a bunch of, some cells are going to have more than others, but a bunch of mitochondria. Mitochondria. And these are found in all eukaryotes. Okay, and these are the sites of cell respiration. Let's say aerobic cellular respiration, which means that the parts of cell respiration that involve the use of oxygen, O2, molecular oxygen is O2, parts of cell respiration that utilize that are aerobic parts of cell respiration. So there's two parts of this, the Krebs cycle and the electron transport chain. These are both housed in the mitochondria. These are processes of cell respiration that ultimately lead to the production of ATP. All right, so what happens is glucose, C6H12O6, with the help of some oxygen that's available in these mitochondria, will get converted into carbon dioxide, which is pretty much useless, and water, both of which are released. And this is the important stuff, 36 to 38 ATP molecules. And ATP is the stuff that is used by cells as sort of an energy currency. In other words, the cell wants to do some work, it has to spend some ATP to make that happen, all right? So basically the mitochondria are the sites where the energy-rich components of the food that you eat will be broken down, oxidized via cell respiration to produce this ATP. So mitochondria are incredibly important, and all eukaryotes, for the most part, possess them, all right? And we now know that they likely are derived from endosymbiosis. They are now a fixture in eukaryotic cells because of that. They provide a benefit to those cells and to themselves that they wouldn't have otherwise. The mitochondria started off as bacteria that were free living and ultimately parasitized larger cells. And that resulted in a mutualism where the two partners did better together than either did separately. Okay, that makes sense. So what do you have in a mitochondrion? And by the way, cells that are really energetically expensive are going to have lots of these. Okay, cells that don't require as much ATP are going to have fewer. Okay, generally they're about the size of a bacterium, because that's exactly what they are. There's going to be an outer membrane. Okay, there'll be an inner membrane in a mitochondrion, which is convoluted. It's folded into these little folds, and these are called cristae. Okay, so this is the outer membrane. And then we've got an intermembrane space, this area in here. And then we've got the inner membrane, which is convoluted and thrown into these folds called cristae. All right. And then in the interior, this place in the interior is fluid filled, cytoplasm filled, if you will. This is called the matrix. All right. And in that matrix, let me use a different color. Use blue. You'll have this little mass of genetic material that if we were to pull that out and look at it up close and tease it apart, we'd find it's a circle. That is the mitochondrial DNA. In other words, it's got its own chromosome with its own genes, and it can code for its own enzymes. And those are what we find either free floating in the matrix or the intramembrane space, or embedded in this cristae membrane. So these are little enzyme proteins embedded in here, well, and others that are used as electron carriers. These are not just enzymes, but they're electron carriers. And because that membrane is folded like it is, you can pack that many more of those in there, and thus can carry out cell respiration much more efficiently than you would otherwise. If you didn't have all those folds in there, you'd have fewer of these proteins and it wouldn't work as well. All right. So that's kind of the structure of the mitochondria. And we know that the two membranes and the mitochondrial DNA are present there most likely because these organisms arose from or derived from what used to be free living bacteria that were taken up by endosymbiosis. All right. So anyway, there's that. And by the way, if we sequence that mitochondrial DNA, we find that it is pretty much the same as bacterial DNA. And we can literally determine which group of bacteria that mitochondria typically derive from. All right. So if you look at an advanced high resolution sort of depiction of relationships among organisms and among bacteria, they'll include mitochondria in there as a distinct group of bacteria. That's how much resolution we can get by studying DNA. So those are mitochondria. All eukaryotes possess them. Now, remember, some eukaryotes are incapable of ingesting their own food, and they have to produce it from other sources. And so those are things like algae, plant-like forms, photosynthetic forms. and they're going to have mitochondria. But in order to obtain the glucose that's needed for cell respiration, the glucose that's going to be oxidized in the mitochondria, they have to produce those from alternate sources. All right. So let me go ahead and erase this because apparently I've reached the end of my whiteboard. Never done that before. Let me erase what I've got here so I can show you the other kind of organelle that's common in eukaryotes that's involved in metabolism. So this next kind of organelle is called the chloroplast. Okay, so this is limited to organisms that use light for the production of glucose. So we'll call these photoautotrophs. And so those would be plants. Okay, plants are eukaryotes that have chloroplasts, and algal organisms, algae. Okay. Fungi don't have them. Some animals actually can harbor algae in their tissues, and thus, in kind of an indirect way, can obtain energy through photosynthesis of their symbionts, the algae that they possess. Things like corals and sea anemones and so on do that. But the organisms that directly, house in their cells chloroplasts are these. And so a chloroplast, again, is likely derived from endosymbiosis. Chloroplasts have an outer membrane, a double membrane. Okay. So there's a double membrane. And then inside, they're going to have, let me use green because that just makes sense. We're talking about photosynthetic organisms and they're green. You'll have internally here these sacs, kind of reminiscent of a Golgi apparatus, flattened sacs stacked on each other. And then you'll have multiple ones of these, maybe several of them. I'll just draw a couple here. All right, so there's a couple of stacks of these little flattened sacs. So each stack is called a granum. And each individual component sac in the granum is called a thylakoid. Okay. And so, and there may be connection through little channels that unite the multiple grana that are present in the chloroplast. So those are some internal structures there. And then there's going to be some DNA. Let me use a different color for the DNA. So here's some DNA in here. Again, this is a nucleoid. That's the chloroplast DNA. It's a circle or nucleoid of DNA. Some people shorthand this and call it cpDNA. They do the same with mitochondrial DNA, by the way. They refer to that as mtDNA. So there's a chloroplast. And in the membranes of the thylakoids, you have the pigment molecules. The pigments include things like chlorophyll and carotenoids that impart yellow or orange or red colors. And those are light gathering. So as light comes in, it's used to drive processes that ultimately lead to the formation of glucose. And so that photosynthetic equation, basically, this is a summary of what happens in the chloroplast. Here we take the CO2, which formerly was a waste product produced during cell respiration. Now it's one of the reactants, CO2 and water, with the help of light, gives us glucose. C6H12O6, and oxygen. That's the predominant way that photosynthesis is carried out in cells. There's other ways that don't lead to the production of oxygen, but oxygen producing photosynthesis is typical of the things that we're familiar with. So we'll leave it at that. But those are chloroplasts, okay? One last thing that I'll mention about the interior of the cell, a feature that they possess that in addition to the nucleus, the endomembrane system, and these organelles, there's also what's referred to as a cytoskeleton. And I'll just put that over here in a box because apparently I can't add any more frames to my whiteboard, but there's also a cytoskeleton. I want to make mention of that. And basically, this is a network of proteins of various sizes, microtubules, even thinner protein filaments, and so forth, that are used to kind of provide a kind of bolstering of the interior structure of the cell. And so it's like if you were to walk into an old haunted house, you know, if the house is like a cell, as you go into a room where, which would be analogous to where the cytoplasm is, spider webs hanging down and going everywhere in there. That's kind of like the cytoskeleton. So it's a web-like framework of protein filaments that are used to maintain the size and the shape of the cell, and sometimes to help it move. So cells, when they divide, they literally change shape. They pinch in two, and the cytoskeleton is responsible for that. So this is a internal framework of protein. And I'm being real brief here with that. So anyway, so that's another feature you'll expect to find. And there's probably some other things I'm overlooking, but like that's a good accounting of the things that you'll find in eukaryotic cells. So with that, I'm going to end this lecture. All right. So I'll be putting together another one or maybe more on the different groups of eukaryotic organisms. So me and this one, and I'll post it, and then I'll get the next one prepared. Thanks.